Hello everyone, my name is Alessandro Busetti and I am one of Metabolon's field application scientists. And today I'm going to talk of metabolomics applications and impact in animal health and husbandry. And in particular, I'm going to present a beautiful publication which touches on a number of very important topics, including animal nutrition, the use of antibiotics, the gut microbiome and its modulation, but ultimately animal health and metabolism. And so the beautiful study I am referring to was published in Scientific Reports in 2019, and the title is In Feed Bacitracin Methylene Disalicylate Modulates the Turkey Microbiota and Metabolon in a Dose-Dependent Manner. And why is this a very important study? Well, in my opinion, it shows how coupling metabolomics with metagenomics analysis improves our understanding of the microbiota's functional output, but most of all of those compounds interacting with the host. So again, in my opinion, this study highlights how microbial taxa associated with beneficial functions can be used as potential targets for gut microbiome modulation to improve animal health production. And how can you modulate the microbiome? Well, you can use prebiotics, probiotics, bacteriophages, but that's another story. And if you want to know more, why not look at my other videos presenting a study on the use of bacteriophages to modulate the gut microbiome. So what's the background to this study? Well, this study stems in part from the ban on the use of subtherapeutic concentrations of most antibiotic compounds for growth promotion in food-producing animals, highlighting the need to discover alternative growth promotions. Now, as you can see in the study design, the researchers wanted to study the effects of feeding a subtherapeutic, so a 50 gram per ton of feed, and a therapeutic, so a 200 gram per ton of feed, concentration of an antibiotic called bacitracin methylene disalicylate, or BMD in short, to commercial turkeys for 14 weeks. And then they wanted to study the effect on turkey intestinal microbial communities and cecal metabolomes. Now, for those of you who don't know, bacitracin is actually a mixture of related cyclic peptides, which are produced by bacteria, in particular the Bacillus genus. And in humans, they tend to be used topically because they cause kidney damage. Now, getting back to the study design in the study was very solid. And in fact, the researchers used 240 male day-of-hatched Nicholas turkey poults, which were initially co-housed to acclimate them for 2 weeks prior to starting adding the antibiotic to the feed. And they were conditioned on a specific litter, which was antibiotic and pathogen-free, to homogenize microbiotas across birds. Now, as you can see in the study design, the birds were fed sub and therapeutic doses of antibiotics for 5 weeks. Then, in the groups that was receiving the therapeutic dose, the dose was lowered to a subtherapeutic dose for another 6 weeks, and then the birds went back on a normal antibiotic-free diet for the last week of the study. Now, birds were obviously euthanized throughout the study, and intestinal and cecal contents were subjected to both metagenomic and metabolomic analysis at regular intervals. And so the first thing that the researchers did was to perform the metagenomic analysis of 3 intestinal compartments: the jejunum, the ileum, and the cecum. Now, both BMD concentrations were found to have an immediate and lasting impact on the microbiota structure. And in fact, they reduced bacterial richness through to the end of the study, so 12 weeks. Now, in the image on the top left, so A, B, and C, you can see the beta diversity shifts of the A, jejunum, the B, the ileum, and C, the cecum microbiome through time as affected by BMD treatment as estimated by the Bray-Curtis measure. Now, the ellipses actually indicate the 95% confidence interval. And as you can see in B and C, the cecal and ileal communities mature quite clearly through the 12-week span of this experiment, while the jejunal community matures to a much lesser extent. Now, on the top right, so in D, you see the alpha diversity of the turkey ceca through time as affected by BMD treatment as estimated by the Chow measure of OTU richness. And the number of OTUs increased about twofold as the turkeys matured from day 2 until day 84 of the study. However, the BMD treatment trended to reduce the species richness throughout the experiment. Now in this slide, you can see the bacterial composition of the turkey jejunum, ileum, and ceca as indicated by genera that compose an average of more than 1% of the community in all gut locations. So other minor genera are not shown. Now, a few important points. All gut locations are actually dominated by genera from the phylum Firmicutes. Jejunal and ileal communities in particular are dominated by lactobacillus. And the microbiota change or maturation through time was influenced, but generally unimpeded by addition of BMD to the feed. And finally, what is evident is that there was a very dramatic switch in the ileal community from being dominated by the lactobacillus at young ages to being dominated by the Rhambutia after day 42. And finally, in this slide, you see some of the results of the Untargeted Global Metabolomics analysis that was performed on the cecal contents. And in particular, you see the counts of cecal metabolites that were differentially present in the animals given the subtherapeutic in the upper panel and therapeutic in the lower panel in feed BMD compared to the control animals. Now, as you can see, similar trends were observed in both the therapeutic and subtherapeutic groups, but to a lesser extent in the subtherapeutic group. Global metabolomics analysis revealed over 700 metabolites in the turkey ceca, and BMD treatment was correlated with changes in these metabolites. Now, of note, the dose-dependent responses to BMD were also apparent in the metabolomic data, so what we saw in the maturation of the microbiome. As you can see, the cecal metabolite profiles of turkeys fed both the subtherapeutic and therapeutic BMD concentrations were different on day 7 and day 35. However, when the diet was transitioned to a subtherapeutic BMD concentration, the metabolite content of the therapeutic and subtherapeutic groups converged throughout the remainder of the experiment. Of the metabolites present in all groups over the course of the experiment, 69% were significantly differentially present due to BMD treatment in at least one time point during the study, suggesting that there is global impacts on microbial functions. And taken together, this shows how BMD exerted a significant short and long-term effect on both the microbiota and metabolomic profile of the ceca, and that a period of recovery to homeostasis in the weeks that follow was also affected. Now, importantly, the BMD affected metabolite profile changed over time. So, for example, at 7 days, 68% of the differentially abundant metabolites were decreased due to the subtherapeutic and therapeutic concentrations of BMD. And mainly, these were metabolites related to host energetics, and nutrient sources, including amino acids, carbohydrates, lipids, nucleotides, peptides, and plant food metabolites. One example of the change in metabolite profile at different time points were dipeptides, so markers of protein metabolism, which have an important role in bacterial host interactions. For example, on day 7, all 17 dipeptides that had been detected trended to be less abundant in BMD-fed poults, but on day 78, the inverse relationship was observed. Now, some metabolite shifts detected in the experiment suggest bacterial inhibition early on in the experiment. For example, on day 7, adenine metabolites were strongly impacted by BMD, so adenosine triphosphate, ATP, deoxy ATP, AMP, ADP, DAMP, increased in abundance due to BMD present in the feed. While some precursors like adenine, adenosine, 1-methyl adenine, 2-deoxyadenosine were all less abundant. Additionally, nearly all the phospholipids were decreased due to the therapeutic use of BMD on day 7. And since phospholipids are a major component of gram-positive and gram-negative cell membranes, this may indicate an overall reduction in intestinal bacterial load. Amino acid concentrations were all also broadly impacted by in-feed BMD, both concentrations. In fact, they were the most impacted category of metabolites in the experiment. And 3 amino acid metabolites in particular, so tryptophan, tyramine, and valine, distinguished both BMD treatment groups from the control group. Tryptophan metabolites were among the compounds that were more predictive of BMD treatment, according to random forest analysis, in both the subtherapeutic and the therapeutic groups. Changes were also detected in indole 3-carboxylic acid, in 5-hydroxyindole acetate, in indole acetate, and N-acetyl serotonin. In fact, tryptophan metabolites are clearly a long-responding group of metabolites in both doses of BMD. And finally, in this slide, we get to the real heart of the study, where I think you can really appreciate the power of being able to integrate the metabolomics and the metagenomics data set to gain a more deep insight on the correlation between specific bacteria and the metabolic output. So, as I said earlier, tryptophan metabolites were strongly predictive of BMD use, and the researchers decided to investigate how the microbiota correlated with their abundance to gain a greater insight in their metabolism. As you know, tryptophan is a precursor of a large number of microbial and host metabolites. The majority of dietary tryptophan is metabolized in the intestinal tract by microorganisms into indole and indole metabolites, indole 3-aldehyde, indole 3-acetic acid, indole 3-propionic acid, indole 3-acetaldehyde, and indole acrylic acid. Now, these microbiota-derived metabolites are endogenous ligands of the aryl hydrocarbon receptor, which similarly to the toll-like receptors regulates immune response and homeostasis at the level of the intestinal epithelium. So the researchers measured a number of tryptophan metabolites, including indole molecules, and found that indole 3-carboxylic acid, indole acetate, 5-hydroxyindole acetate were among the top metabolites distinguishing both BMD doses from the control birds. And to date, only a few specific bacteria are actually known to produce endogenous ligands of the AHR receptor, which affect host physiology. And in the data set, they identified bacterial taxa that correlated with tryptophan abundance, and 3 families composed most of the species correlated with tryptophan metabolite concentrations. And these were the Clostridiales, the Lacnospiraceae, and the Ruminococcaceae. Now, it is well known that Clostridium sporogenus, a member of the Clostridiales, metabolizes tryptophan to IPA and IAA, and that both these molecules modulate intestinal permeability and immunity. In the ceca, BMD use in the feed increased the amount of IAA, 3-indoxyl sulfate, 5-hydroxyindole acetate, as well as decreased indole 3-carboxylic acid at the end of the experiment, so towards day 78 to 84. Now, in humans with inflammatory bowel disease, there is an inverse correlation of tryptophan metabolism and production of endogenous AHR ligands and severity of clinical disease. So, for example, humans with ulcerative colitis have less IPA in their serum compared to healthy age-matched controls. So the increasing the levels of IAA and 3-indoxyl sulfate may actually have a very beneficial effect to the host. So again, this, I hope, highlights how modulating the microbiome with this antibiotic leads to beneficial traits that can be targeted to improve animal health and production. So a very quick summary slide highlighting some of the major results of this study. So as expected, both antibiotic concentrations had an immediate and lasting impact on turkey's microbiota structure and reduced the bacterial richness through to the end of the study. Metabolomic analysis was able to identify 712 biochemicals, 69% of which were differentially present in BMD-treated turkeys for at least one time point in the study. These included amino acids, carbohydrates, nucleotides, peptides, and lipids, which were decreased in turkey ceca early in BMD administration. As we saw, there were some long-term alterations in the metabolon, which continued even after the withdrawal of antibiotic treatment. The microbial composition, which was determined using 16S rRNA gene sequencing, was predictive of the metabolon, indicating a connection between the microbiome and the metabolon. Finally, as I said, in-feed bacitracin methylene disalicylate, or BMD, causes bacterial metabolic shifts, leading to beneficial traits which can be targeted to improve animal health and production. Now, thank you all very much for your attention. I do invite you to contact Metabolon if you would like to know more about not just this study, but how we can come and help you in your specific research area and help you overcome some of the daily hurdles which, as investigators and researchers, we all face. Thank you very much.